Friends, Namaste. Welcome to Dr. Varsha Gaitonde's channel. In this video, we shall talk about the concept apomixis. Along with apomixis, we shall also discuss some of the topics or concepts about the sexual behavior or reproductive process in the angiosperms. Before going into the topic, we shall see some of the ploidy levels in different parts of the reproductive parts of a plant. So, the haploid condition is generally observed in microspore or pollen, sperm, tube nucleus, immature embryo sac, megaspore, egg cell, ovum, synergids, antipodals, polar nuclei, most fungal spores, hyphae of most fungi, and yeasts. Similarly, the diploid or 2N condition is observed in zygote, embryo, ovary, ovule, integuments, pericarp, nucellus, stigma, style, secondary nucleus, megaspore mother cell, pollen mother cell or microspore mother cell, anther, filaments, anther wall, anther before microsporogenesis, petals, sepals, thalamus, flower pedicel, flower bud, plumule, radicle, seedling, stem, leaf, leaf tip, midrib, petiole, root, root hair, root tip, root cap, and shoot tip. This is nothing but the root tip is nothing but radicle and shoot tip is nothing but plumule. That is the covering portion in order to protect it. The triploid condition, 3N condition, is only observed in three structural parts, that is primary endosperm nucleus, aleuron, and endosperm. Now let us label the mature ovule, we may say, at the ovarical part. So whenever we talk about the female reproductive part, if we talk about the self-pollination here, the same anther or the same male portion will fertilize the female part, whereas in cross-pollination it should come from the another flower. Okay, but in self-fertilization it is from the same flower. Okay, so whenever either it may be self-pollination or cross-pollination, when we talk about the anther portion, we will find the anther and filament, that is in male reproductive part. So whenever fertilization has to happen, what will happen? From the anther, one cell will enter here, or we may say the male gamete will enter here and it will travel towards the micropylar end or chalajal, and it again depends on the different species or different genera. So whenever this particular pollen is traveling from here to here, again it will go to the mitotic division and divides into two nuclei, that is, one is vegetative nuclei and one is generative nuclei. So after this division, that will enter into the micropylar end, and what will happen? Here already the condition is 8 nucleide in the embryo sac. Inside the embryo sac, the 8 nuclei stage will be further divided into 3 synergids here. Sorry, 2 synergids this side, 3 antipodal cells at the other end, that is chalajal end, and 2 polar nuclei at the center. So whenever the male nuclei will come, it will start fertilizing. It will first, before fertilization, fusion should happen. So first it will fuse with one egg nuclei and another with, because in the division already two nuclei formed, one is vegetative and one is generative. So one will fuse with the polar nuclei and another will fuse with the egg cell. So what will happen here? Here the double fertilization process takes place, and whenever by fusing this 2N condition, and that is antipolar nuclei and one egg, one vegetative nucleus or generative nucleus, whatever it may be, will fertilize here. That will become a 3N condition to form the endosperm. That condition is called as triple fusion. So let us see the other parts of the ovule. the embryo sac. This is embryo sac, that is immediately after that the outer covering is inner integument, this one. Then there will be one outer integument at the one end. At micropylar side, hilum and funicles will be formed. Here the funicle is the attachment portion which holds or give a structural property to this particular ovule. Then the other parts like vascular strands, then nucellus, the outer cellular portion, synergids, synergids and antipodal cell, all these things I have already labeled. So let us move to the actual process of reproduction. So the mode of reproduction can be divided into sexual and asexual process. First, we shall talk brief about the sexual process. In sexual process, what will happen? There are three conditions, generally autogamy, allogamy, and often cross-pollination. Autogamy is nothing but self-pollination, as I have already told that the pollen of same flower will pollinate the stigmatic portion. Then allogamy means it is in the reverse order, that means from the different flower it will pollinate on the different stigmatic portion. And often cross pollination means where the cross pollination is observed as 5 to 30%. Now we shall talk about the asexual reproduction. Asexual reproduction is further divided into vegetative and apomixis. Vegetative reproductive process is again divided into natural vegetative reproduction and artificial reproduction. Natural reproduction means in nature whatever happens by asexual means. That may be because of the difficulty in pollination habit or fertilization or difference in the ploidy level and all such conditions. Whenever the perennial trees are facing difficulty in flowering or the fertilization process, generally they will undergo the vegetative reproductive process. So here it is further divided into underground stem and subaerial stem. Underground stem is further divided into rhizomes like turmeric, ginger. In all these plants, rhizomes are the propagating materials. Tubers are the propagating material in potato, corm in arvi and banda, then bulbs in onion, garlic. Subaerial stem. This is observed as runners in strawberry, stolons in rhodis grass, then suckers in banana, and bulbils in garlic. Along with this, roots are also the propagating material in many of the vegetables, like swollen root can be observed in case of beetroot and all those things, arrowroot, like that. And now we shall discuss about the artificial vegetative reproduction, like stem cutting. In stem cutting we practice this in sugarcane, grapes, rose, etc. Root cuttings are obtained from sweet potato, citrus, lemon. Layering. Generally, it is practiced as air layering, that is found in guava and fruit crops. Then grafting. Grafting generally done in forest species. Then guti. Guti is nothing but rooting of the air layered plants, that is practiced in apple and guava. Then budding is practiced in birch, maple, dogwood, redbud, then ginkgo. Coming to the process of apomixis. Apomixis is nothing but it is the formation of embryo without sexual fusion. Asexual fusion was because of different means by cutting, budding and all those things. But there is another process where inside the cell, that means not physically, not visibly, but inside the flowering portion or flower portion only, some things happen. Because of that, apomixis is the famous process where the sexual fusion or undergoing the meiosis can be prohibited at the initial stage. So what will happen here? Here the embryo forms without sexual fusion. When we talk about apomixis, there are two relevant other terms. One is parthenogenesis and another one is parthenocarpy. Parthenogenesis is a condition where the embryo is formed from the egg cell. Egg cell I have already labeled when we talk about the 8 nuclei condition inside the ovule. Here nearby the synergids one egg cell is found. This middle portion is the egg cell. If the embryo is directly forming from this and without fertilization, that is known as parthenogenic condition, whereas parthenocarpy is, that is the formation of fruit without seed formation. Okay, that is parthenocarpy. So now we shall see the broad classification of apomixis based on frequency, based on occurrence, based on the reduction division, and based on the cell division. It is classified. Coming to the first one, based on frequency. Based on frequency, it is divided as obligate apomixis and facultative apomixis. Obligate apomixis means the apomixis by the only means of asexual reproduction, whereas facultative apomixis means here some portion of sexual process also happens, or sometimes it may undergo the sexual process or sometimes into asexual process. Such apomixis is known as facultative apomixis. Then coming to the based on occurrence, the division is classified as recurrent apomixis, adventative embryonic, and non-recurrent apomixis. Recurrent apomixis is further classified as apospory, diplospory. Example, diplospory is the embryo formation or embryo sac development from the megaspore mother cell. Then adventative embryony, which is also called as sporophytic budding. Then non-recurrent apomixis. This is further divided into haploid parthenogenesis and haploid apogamy or pseudogamy. Haploid apogamy is also called as pseudogamy. What is apogamy, apospory and all, we shall discuss later. Then the third kind of classification is based on the reduction division. This is classified as direct initiation of embryo sac and unreduced somatic cells. Direct initiation of embryo sac is also called as sporophytic apomixis. I shall tell you some of the points regarding direct initiation of embryo sac. In this process, it is also known as adventative or adventitious embryony or adventative embryony. Here, embryo arises from nucellus or integuments of the ovule. Embryo development is initiated as a bud-like structure through mitotic division of the cell nucleus. Example, in case of citrus, remember here meiosis is not happening, only the mitotic process is taken into account. The second one is unreduced somatic cells. This is also called as gametophytic apomixis. Here the unreduced cell give rise to a megagametophyte, then gametophytic apomixis occurs. This is further divided into apospory and diplospory. The fourth kind of classification is based on the kind or number of cells involved in the apomixis process. That is further divided as apomictic embryo sac and apomictic embryo. Remember, based on the number of cells involved, the process of apomixis is divided as apomictic embryo sac and apomictic embryo. First we shall see the apomictic embryo sac. This is further divided. This is also called as apospory. Apospory is the condition where the diploid cells of the ovule lying outside the embryo sac develop into another embryo sac without reduction. Here the embryo sac picture is already shown. With the help of this diploid embryo sac only, or the cells lying outside this embryo sac, another embryo sac is developed and there won't be any kind of reduction. Always remember that apospory always occur in diploid condition. There won't be like haploid apospory. Okay, only diploid apospory happens. That's why here I have eliminated that haploid condition. Example, in case of Parthenium and Crepis, apospory condition is found. Coming to the second classification, that is apomictic embryo. Here endogamy, apogamy and parthenogenesis are the three conditions. So we shall see one by one. Androgamy. Andro means, the name suggests, andro means the male reproductive part. Embryo develops from the embryo where the male embryo is taken into account. That means if it is developing from either the pollen coming from the or the male part of that particular flower, that is nothing but endogamy. Here no ovule or none of the embryo sac pore parts or nuclei is involved in the process of developing an embryo. Second is apogamy. Apogamy is nothing but development of embryo either from synergids or antipodal cells. That means the cells which are adjacent to egg cell or the middle one, that is antipodal cells. These things will give rise to apogamy. For your better understanding, I'll show it in the picture. Here, from the antipodal cells are at the other end. They are 3 in number, and another 2 synergids, means from the 2 different poles the cells are taken or considered for the fertilization. That is nothing but apogamy. That is further divided as haploid apogamy, diploid apogamy. Haploid apogamy is embryo develops from the haploid synergids or antipodal cells, and diploid means it is development of embryo from diploid synergids or antipodal cells. Example, in Allium species. The third one is parthenogenesis. That is development of embryo from egg cell without fertilization. Further, parthenogenesis is divided into haploid and diploid. Haploid parthenogenesis means unfertilized egg cell developing the haploid embryo. Example, in Nicotiana, that is tobacco family and maize family. Then diploid condition is diploid parthenogenesis is diploid embryo develops from diploid cell. Example, many grasses like Taraxacum. This was all about the classification of apomixis, and along with that we also differentiated the sexual and asexual process. Now in case of apomixis only one more condition is there, that is known as syngamy. It is the condition where haploid sperm cell nucleus enter the egg but doesn't fuse with the haploid egg nucleus. Here each nucleus divides independently, creating a haploid embryo that contains sectors of male and female origin. That means fertilization won't happen, but pollination is essential for the development of particular embryo. That is, the condition is known as syngamy, and this condition is observed in cotton. Coming to some of the basic information about apomixis. Apomixis is a Greek word taken from a two-word fusion, that is apo means away from and mixis means act of mixing or mingling. The first discovery of the phenomena credit goes to Leeuwenhoek in early 1790. About in 300 species belonging to 40 different families, apomixis process is found, generally found in polyploid species like Pova, Paspalum, Panicum, Panicetum, Cenchrus, Citrus, Malus, Bulbus, Mangifera, Gia, and Beta vulgaris, that means maize and beetroot family. The process is most frequently observed in Compositae, Rosaceae, and Graminae family. Remember, apomixis has not played any evolutionary significance so far. That means apomixis is a an advantage to one generation only. That means over a period of time, none of the differences are observed in the different generations or in heredity or evolutionary process. This has not played any role. Associated with perennial cross open pollination or interspecific hybridization along with polyploidy condition. The genotype of the embryo and resulting plant will be the same as seed parent, just like clonal propagation. Genotypes of developed plants are identical to the parental types, just like clones. Now we shall see, if any plant is undergoing apomictic condition, how to detect that particular plant. There are different techniques like phenotypic analysis, cytological or cytoembryological study, microbiological method, or marker assisted selection or marker technology used in order to detect the apomixis process. Combination of these methods can also be used for the better results. We shall see one by one. First one is phenotypic analysis or identification. Here, the phenotypic classification may involve least work of any technique for detecting apomixis, but is also least reliable. Codominant or incomplete dominant alleles are necessary for proper classification of the heterozygotes. Phenotypically, whenever the process of embryo is deviating or the development of polyembryonic kind of condition, we shall easily detect that something it may be apomixis. Second technique is cytoembryological detection. Here the observation of developing spores and embryo are done. This can be quite laborious but does enable the detection and classification of form of apomixis. Other methods like auxin test, cytological test, ovule culture test. Auxin test can be used to demonstrate the estimation of the frequency of apomixis in Kentucky bluegrass, that is a facultative apomictic crop. The molecular marker technique is the third method. Here, apomictic mapping and identification of the region containing apomictic gene was done in the South American grassland species of Bracharia documents segregating for reproductive mode among its progeny. The researchers mapped the clones of maize chromosome 5, 1 to 5 centimorgan from either side of the apolocus, means apomictic locus. The apomictic genes in the wild plant species, that is Panicum squamulatum, have been successfully transferred over into the cultivated pearl millet species Panicum glaucum following 3 generations of black crossing with the 2 plant species. That means by using the molecular markers, apomictic condition was also transferred, or it was just observed from wild into the cultivated species transferring process by using the flanking markers. Now we shall discuss about the genetics of apomixis. The genetics is further divided into based the classification or the process based on genes involved in female meiosis and reproduction, that is observed through genes controlling meiosis, genes controlling megaspore mitosis, genes controlling embryo and endosperm development. Then modifications observed in apomictic species, that is modification or failure of meiosis during megasporogenesis, initiation of mitosis in the unfertilized 2n egg cell or some other 2n egg cells of embryo sac. Other 2n cells of embryo sac, not the egg cell, means along with egg cell, whichever in the diploid condition, those cells can also be considered. Then in apospory and adventitious embryony, there is initiation of mitosis in vegetative cells leading to embryo sac or embryo development. Then the third classification is number of genes controlling, that is single gene or locus like in penicetum, panicum and citrus, dominant over sexual reproduction in case of panicum, citrus, single gene with other tightly linked genes in trypsacum, and apomixis controlled by 4 recessive genes in Parthenium argentinum. Now we shall see how to develop an apomictic line if we are intentionally imposing the apomictic condition in different plants or a particular plant. So first we should do the gene transfer from wild species. This is quite different. The genes controlling apomixis can be transferred into a crop species from related species, like the approach is already practiced in Tripsacum dictyloides, that is an wild ancestors of maize, and from there it is transferred into maize. Then from Pennisetum orientale, that is a wild ancestors of bajra, and that is transferred to Pennisetum species which is cultivating. Then from beta trigena to beta intermedia Elymus species into the cultivating Elymus form. Then induced mutations. This approach aims in developing apomictic forms in normally sexual reproducing species by utilizing induced or even spontaneous mutations. These efforts have focused primarily on sorghum, where two mutant lines showing facultative apospory have been isolated. Example, sorghum line R473. Third approach is isolation of apomictic recombinants from interspecific crosses. In this approach, sometimes apomictic recombinants can be recovered from segregating generations of crosses between two sexually reproducing species. Example, seeds formation has been reported in intergeneric hybrids between Triticum estimum and Avenus sativa, then Hordeum vulgare and Triticum turgidum, like that. Then engineered apomixis. In case of engineered apomixis, by making the artificial operon, means by designing the different parts of the operand, like how it should be, like which kind of operator gene, which kind of promoter gene, or which kind of repressor, along with structural gene combination, how the process of apomixis can be regulated by a cassette, designing a cassette, that is nothing but the genetic engineer or engineering apomixis. The role of apomixis in plant breeding, or we shall discuss the applications of apomixis. First one is a rapid production of pure line or homozygous lines. The parthenogenic development of reduced egg cell followed by reduplication or cell fusion or development of embryo from secondary diploid cells of the embryo sac derived through fusion. This is observed in rubus and sorghum. From true breeding sexually reproducing progenies, which are identified in F3 generation of crosses R473, which is an apomictic line of sorghum, and that is crossed to a sexual line, like three different lines, and the fusion of haploid nuclei of embryo sac has been cytologically verified. That means this yielded the apomictic, which are homozygous or pure lines. Maintaining the superior genotypes. Apomixis is useful in maintaining the characters of mother plant. That means whenever we are interested in one particular line, and to maintain that particular superior genotype without altering its genetic constitution, then we may go for apomixis approach. The third process is conservation or fixation of heterosis. For this, apospory or diplospory condition is generally preferred. In some cases, hybrid vigor may be conserved for many generations by using recurrent apomixis. Sexual reproductive line, that is used as female, and apomictic line is used as male, and by crossing this, apomictic hybrid is generated, and that is maintained and multiplied through seed production or normal sexual process. That is how the conservation or fixation of heterosis is observed through apomixis process, or that is achieved. Production of hybrid is the fourth approach. Hybrids are nothing but the crosses or the combination of two facultative apomixes. Remember, two facultative apomixes. Facultative means that may follow sexual as well as asexual process. Why we are talking about facultative apomixis, why not obligate? Remember, obligate apomixis is only through the asexual means. That means when the apomix is obtained from asexual only through asexual mean, in the next generation, if it is not crossable, we won't get the favorable results, or in otherwise we may say to alter in our own way in the further generations, facultative apomixes are most preferred. Coming to maintaining the vibrates, how the vibrates are maintained: harvest the seeds of only F1-like apomictic lines in each generation. In sorghum, vibrates preference performance is in between hybrids and pure lines. Remember, vibrate performance is in between hybrids and pure lines. This was observed in sorghum. How to maintain these pure lines? Vibrates require selection in F1 type plants. Then seed certification will produce difficulties since vibrate population will comprise both apomictic and sexual progeny. Then commercial utilization is still under question. Coming to the idea of apomictic species, that means how an ideal apomixis system should be. All the progeny of plants should be apomictic so that progeny have the same genotype as the maternal parent. Example: obligate adventitious embryony, somatic diplospory, obligate diplospory follow this kind of condition. Here no chromosome pairing or recombination and restitution occur at or just after the division of meiosis. That means three processes are forbidden here, like chromosome pairing or recombination, then restitution, that should occur at or just after the division of meiosis. The second condition is apomictic genotype should preferably be fully male fertile or self incompatible and reproduce via pseudogamy. This statement is very important, that is, apomictic genotype should preferably be fully male fertile and self incompatible. That means male should be there but the plant is self incompatible, that is, unable to pollinate the same pollen from the stigmatic portion of, or it will pollinate but the germination and other processes are forbidden here. Pollen production is essential for the transfer of gene controlling apomixis to normal sexual genotype. In case of diplospory, chromosomes should not pair or recombine during first meiotic division, which may give rise to variation among the progeny. If recombine, it may cause variation in progeny. Means in a diplospory condition, the chromosome should not pair or recombine during first meiotic division, that is the reductional division. If it happens so, the variation will observed in the progenies. In case of diplospory only, restitution should occur at this after the first meiosis. If it occurs after second division of meiosis, little or it is of no use in plant breeding because again it creates the variation. Apomixis should be dominant over sexual reproduction. Usually, apomixis is governed by two or more genes. Expression of apomixis should be little influenced by the environment, or else we may not find it in the further generations. As I have already told, apomixis played no or very little significance in the evolutionary process. Coming to the advantages of apomixis over male sterility and self-incompatibility, the technology is relatively sophisticated than farmers. Here, the disadvantages of male sterility and self-incompatibility are highlighted. The male sterility and self-incompatibility technologies are relatively sophisticated, but farmers has to buy fresh seeds every year and the two parents of the hybrid must flower at the same time. Coming to the advantages of apomixis in breeding, rapid multiplication of genetically uniform individuals can be achieved without risk of segregation. This is just a kind of process of cloning like. Heterosis or hybrid vigor can permanently be fixed in crop plants, thus no problem for recurring seed production of F1 hybrids. Efficient exploitation of maternal effect, if present, is possible from generation to generation. Then homozygous inbred lines, as in corn, can be rapidly developed as they produce sectors of diploid tissue and occasional fertile gametes and seeds. The fifth advantage is obligate apomixis permits fixation of heterosis in the hybrids, so the farmer can resow the seeds after produced and apomictic hybrid generation to generation they can maintain. The new hybrid variety could be multiplied from few hybrid seeds in the same manner as pure line. This greatly simplifies hybrid seed production. Even such parents that flower at different times may be crossed in the greenhouse to obtain few hybrid seeds, which can be used to establish the new hybrid variety. Nucleus seed of hybrid varieties can be conveniently maintained as hybrid varieties. Hybrid seed field requires minimum 3-meter isolation only to prevent mechanical mixtures, and pollen dispersal is not relevant during hybrid seed multiplication. That means for male sterility, or whenever we will go for the hybrid seed production by following either the male sterility or self incompatibility. Remember, self incompatibility process is very rarely used in the hybrid seed production process. So whenever we are using male sterility or by artificial means, we should follow certain norms like keeping the isolation distance, means spatio-temporal isolation is must, but that is minimum in case of if we follow the apomictic process. One more thing is when we talk about the rice apomictic lines, it is also called as one line system where maintaining the lines is not at all a problem. Coming to the utilization of apomixis, apomixis is very complicated phenomena. Its utilization necessitates reorientation of breeding procedure and seed certification. Estimation of the level of facultative apomixis, which is tedious and time consuming. In case of facultative apomixis, the proportion of sexual progeny is affected by environmental factors like day length, etc. Short day promotes apomixis in Diacanthium aristatum, but it encourages sexuality in Pova ampalda and temperature. Then, in the absence of morphological markers linked with apomictic development, maintenance of apomictic stock becomes a difficult process. Genetic basis of apomixis is not clear in most of the cases. Now we shall see the difference between apomixis and amphimixis. The term apomixis was coined by Winkler in 1906 and amphimixis was coined by Wiesmann in 1891. As I have already told you, the process of apomixis was observed in by Leeuwenhoek. Second difference is sexual fusion or union of male and female gametes. It is not there in apomixis but present in amphimixis. Gene combinations or gene flow is not possible through apomixis, but it is possible in amphimixis. Segregation occurs in apomixis, not in amphimixis. Then rapid development of inbred line is possible through apomixis, not through amphimixis. It needs around 6 to 7 generations. Then conserving heterosis is possible in apomixis. Type of reproduction is asexual. Then in amphimixis it is sexual. Different kinds of apomixis are parthenogenesis, apogamy, apospory, adventitious embryony and all. But in amphimixis it is autogamy, allogamy or often cross pollination. Frequency in plants is low in apomixis and it is high in amphimixis. Now we shall see how the apomixis is different from different systems of hybrid seed production. Whenever we talk about the hybrid seed production, what we will the different methods exploiting are male sterility. If we talk about the two line system, that is CMS requires at least two lines. One is the maintainer line, means A line and B line are required in case of CMS maintenance. When we talk about the three line system, just see here, this is two line system. Here what will happen when we talk about gametophytic apomixis, 50% of the plants need to be discarded every year. That means that will cause a huge loss. If I talk about three line system, here A, B, R line are followed. Then restorer line is also taken here. Maintaining the restorer line is very difficult, and when we go for three line system, the space and time consumed for the process of seed production is quite high. That is laborious as well as costly approach. Another two things we follow like protrandy and protogyny condition. When we talk about the maize, we follow the protrandy. That means androecium will mature first by removing all the tassel portion. What we'll do, we'll achieve the uh, only will achieve like we will remove all the male parts, and so that we can achieve the cross pollination from whichever the desirable pollen source we are having. Then protogyny, this condition is preferred in case of bajra, where the pollen production is quite large but the pollens are very small. Protogyny means the female mature first and male later, so the male portion is removed and female is encouraged. Likewise, in case of apomixis, what we will do? We will produce an F1 by crossing two parents, and we may maintain the same F1 parental constitution for different generations. That is the beauty of apomixis. By using a single line, we may generate for different generations. So here I have added some of the additional information. There are different steps in determining the mode of pollination, like examining the flower for dioecious. Dioecious means the condition where male and female reproductive parts are situated on different plants, not on the same plant. Monoecious means on different same plant. Then protandry, androecium matures first, protogyny, gynoecium. Then cleistogamy is closed flower condition, which is observed in wheat and such kind of self-pollinated crops. Cleistogamy promotes self-pollination, and dioecious, monoecious, protandry, protogyny promotes various degrees of cross-pollination. The second mode of determining the mode of pollination is growing plants in isolation in different locations. If seed set is occurring, that is cross-pollinated; if not, self-pollinated. Then selfing the seeds by bagging. If inbreeding depression is observed, that means it is cross-pollinated, and if not, it is self-pollinated. Coming to the amount determining the amount of cross-pollination. For this, grow plants with dominant and recessive traits in mixed stand and allow open pollination. Dominant and recessive characters means we can phenotypically observe the characters, and by experience we may go for this. And a combination of dominant and recessive characters in a single population, what we may do, scoring the frequency of dominant phenotypes in the progeny of recessive plants, then proportion of dominant phenotypes in the progeny would indicate the extent of cross pollination. Here the black dot indicates plant with dominant traits like red flower, then plants with recessive traits like that, these are white flowers. Coming to detection of apomixis. From the same population, grow plants with dominant homozygotes and recessive traits in a mixed stand and use recessives as females. Means recessive plant should be females. Again, score the frequency of dominant phenotypes in the progeny of recessive plants. Then absence of dominant phenotypes in the progeny would indicate the presence of apomixis. Now, finally, there are some breeding processes followed for asexual propagation of the species, like plant introduction, clonal selection, mass selection, heterosis breeding, mutation breeding, polyploid breeding, distant hybridization, transgenic breeding, then genetic engineering, etc. Mass selection is rarely used in asexually propagated crops. Friends, so far we discussed about the reproduction in plants, how the apomixis, its classification, then the detection of apomixis, its advantages, disadvantages, then along with that pollination habits, like how to detect cross pollination, how to detect apomixis, how apomixis is better than male sterility and self incompatibility. All these things we studied. If you have not understood any of these things, just put it in the comment box so that I may find a better answer for you. And thanks for the patience listening and watching. Thank you once again.